Hello, welcome to another lesson from the conscientious biologist, Ben Gallagher. Now, hopefully you've been watching these roughly in sequence because this one's Digestion 1, which is an introduction and process overview. But it's really quite important that you've watched at least the proteins playlist. So that was 3 lessons, the second 2 of which concern enzymes, and enzymes are a major part of digestion. So please do make sure you've watched those 3 proteins lessons before you come to this one. This one's aimed at GCSE level, and as always, please do subscribe to my channel if you haven't done so already. So let's start digestion with a relatively straightforward question of what is it and why is it important? But before we can answer that, we need to look at what the cells need, because really digestion is just about providing for the cell's needs. So up above me there, from the cell biology lessons that hopefully you've watched as well, we've got a picture of an animal cell. That's a eukaryotic cell. That's really what we're focusing on when we're talking about digestion, because we're looking at the human digestive system. Though of course the human digestive system is virtually identical to all mammalian digestive systems. That's all mammals. But let's think about what cells need if they're going to function. So think about what they do. So starting off with, we need glucose, we need amino acids, we need fatty acids and glycerol, and we need nucleotides. If we consider why though, because of what the cell does, glucose is needed for respiration because we react it with oxygen to get the energy back out of it. So glucose is an energy source for the cell. Amino acids, if you've watched the protein lessons, you would know that they're the building blocks of proteins, and proteins do everything. So we need to get a lot of amino acids to our cells. We need fatty acids and glycerol because you can stick those two together to form lipids. Lipids are an energy store, and they're the building blocks for membranes. So if the cell's going to grow, it needs more membrane, it needs more lipids. And then nucleotides. Nucleotides are the building blocks for DNA. So you can't make more DNA, which remember is an essential thing that you have to do before a cell can divide, so before it undergoes mitosis. You need to double the quantity of DNA, so when you divide it into 2 new cells, each cell has a full set of DNA. So cells need glucose, amino acids, fatty acids and glycerol, and nucleotides. There are some other things as well, but those are the ones I want you to focus on for digestion. Now the reason why we need those is we can't make those things ourselves. So we need to get them from other organisms. To do that, of course, we need to eat those organisms because those organisms are already using those nutrients as part of their own cells. If you're eating a beef burger, well, the beef came from a cow. The nutrients that are inside the beef were from the cow's cells. They're the cow's proteins that were carrying out the cow's metabolic functions, the cow's DNA inside its cells, the cow's carbohydrate stores to get your glucose, the cow's membranes and fat reserves to get your lipids. So those nutrients were being used by other organisms in their own cells. So we need to eat their cells to get those nutrients. Now some people go, well, I don't eat cells. Well, you do, because almost all our food is cells. Because think, we eat plants and we eat animals. There's very few things in our diet that aren't plant or animals, or at least refined from them. So if you're eating anything that contains sugar, that sugar is likely to have been processed from starch from a plant. Things that have been manufactured like chocolate and things like that, that's taken ingredients from cells to rebuild stuff. But most of the food you eat, vegetables, fruit, meat, things like that, they're just cells you're eating to get those nutrients. Digestion is how you get those nutrients from that cellular material. So this one's probably the most important slide of this video. Let's look at the actual process and principle behind what digestion is. So we've already said that we get our nutrients from eating the cells of other organisms, and inside those cells will be the polymer molecules that those organisms have been using. So over there you'll see I've created a polymer. I've stuck a load of those little beach ball symbols together. That polymer would be loads of the units stuck together. So that might be a protein where the units, remember, are amino acids. It could be a carbohydrate where the units, each one of the beach balls, is representing glucose. It might be DNA where each one of those units is representing a nucleotide. But those are polymers, poly meaning many, like the polypeptide that we talked about with proteins. Now, examples of those polymers are, of course, carbohydrates, proteins, lipids, DNA. Those are the things I've just mentioned. Now, we've got to break those polymers down into monomers. And all that means, if you look at what's going on up there, it's taking all the stuck together pieces and breaking them up individually. Monomers are literally the building blocks of what you use to make the polymer. So for carbohydrates, the monomer unit is glucose. For proteins, it's amino acids. For lipids, it's fatty acids and glycerol. Each lipid is made of a glycerol and a couple of fatty acids. And DNA is broken down into nucleotides. That's what you build it out of. Now that process, that breaking down polymers to monomers, that is digestion. That's it. It's that straightforward. Okay. You need to make sure you remember these examples, but basically that's what digestion is. We can add a why do we do that onto this though. Why do we do digestion? It's so that we can get those monomer units small enough so they can pass out of the small intestine by diffusion or active transport. If you remember, if you go to the transport through membranes lessons, you'll see you can recap and review all of that. But they'll move out, they move into the blood. So that's now the transport bit. They've got to be small enough to be transported around in the blood. Then they've still got to be small enough to diffuse into the cell that requires it, so through the membrane, so that that cell can use it as the building block to build its own stuff. So it's got to be small enough to absorb, transport and use. So that's why we do digestion. Okay, we can simplify that if we link it up to what we did in the proteins lessons, where we said about the building blocks being like the monomer units. If you've got a house built out of those building blocks, and we pretend like that's a house in another organism, what you need to do with digestion is break it back down to the individual building blocks so you can absorb them, transport them, send them to your own cells to build them into whatever you want. So that's the kind of overall how and why. But we need to be more focused in these videos just on the how. Adding to this slightly, just on that digestion arrow, you'll see that I've now put in red physical and chemical. That's the how. Okay, to break down polymers, you can do it physically or you can do it chemically, and that's what we're going to look at next. So of these two forms of digestion, let's look at physical first, because that's the quickest one. So physical digestion are processes that physically smash up the food into smaller pieces. They don't necessarily break the chemical bonds down to create the monomers, but it's the start of the process, breaking the bigger bits into smaller bits. And there's two main ways that you do this. The first one is chewing. Now the correct name for chewing is mastication. Please be careful with your spelling and pronunciation of that one. Chewing uses the teeth, of course, uses the tongue to move the food around, and it physically smashes the food down into smaller bits. Now you can tell a lot about an animal by the teeth that it's got. So for example, for us who are largely omnivorous, we've got these incisors at the front. They're the sharp slicing teeth, so we use those largely for cutting through meat or slicing through tough vegetation. You've got the canines. These are the big fang like ones, not very big in us because they've kind of de-evolved a bit. Canines are only for predators who are catching live prey that they need to sink their teeth into that living creature, so they need big canines to sink in. We don't need those, but if you see an animal with big canines, you know it's a carnivore. Our most important ones, really, same as most herbivores, are the big molars at the back that we use for grinding tough vegetation down. But no matter what teeth you've got, whatever the animal is, chewing is the first step to breaking down your food by physical digestion. The second form of physical digestion is churning. This is something that happens once food gets into your stomach. So you chew the food up, you swallow it, gets to your stomach, and if you look at the picture of the stomach above me, you can see all those lines inside it. Those are lines of muscle. So those muscle fibers all contract and make your stomach do that kind of mashing, squelching thing that you can feel happening inside you. That pulping, that churning, also physically breaks down the food and turns it into a bit more of a mush. That, of course, increases its surface area, meaning there's more surface for the chemicals can attack. You really need to have watched the surface area to volume ratio presentation for that, which is also in the Transport Through Membranes playlist. Let's move on to chemical digestion now. Now, chemical digestion is what does most of the breakdown of the polymers to monomers, because this uses chemicals and biomolecules to break chemical bonds. Now, let's look at those main chemicals. The first one should be hopefully obvious to you. It's the hydrochloric acid that you've got inside your stomach. So that acid is really quite strong. So when the food goes down into your stomach, you've got the churning to break it down, but the acid in your stomach is also dissolving it and breaking it down into smaller units. Your stomach acid also has a role for your immunity in that any pathogens, that's harmful bacteria, fungi, viruses you might swallow, will hopefully be killed in the stomach acid as well. Moving on to the most important type of chemical breakdown though, and this is an example of a biomolecule, biologically produced molecule, you've got your enzymes. Now hopefully you've come to this presentation straight from the proteins lessons. So you've just learned loads and loads of stuff about enzymes. But let's look at the main enzymes by looking at the main polymers that they break down, the main substrates, and the monomers that they create. So we know we've got carbohydrates, proteins, lipids, and DNA. Those are the main polymers that you eat. Those all get converted into glucose, amino acids, fatty acids, and glycerol, and nucleotides. But the enzymes that do that process, and you might see a pattern here, are called carbohydrase, that breaks down carbohydrates, protease, that breaks down proteins, lipase, that breaks down lipids, and DNA, which is actually nucleic acid, is nuclease. So the pattern there makes it really easy for naming enzymes because it's the thing they break down, but you take the ending off and you replace it with ase. So if something ends in ase, you know it's an enzyme. Now I should point out all of these are just families of enzymes. There's loads of carbohydrases, loads of proteases, and so on. There's only really one specific example you guys need to know for GCSE, and that's one of the carbohydrases, which is amylase. Now, amylase is the main enzyme that breaks down starch, which is the main carbohydrate that we get our sugars from. Starch is the polymer of glucose that's made by plants. So when plants do photosynthesis, make their glucose, they stick it all together in big polymers called starch. Now, you might be thinking, well, why isn't it called starchase then? And it's because the name of the enzyme is from the proper name for starch, which is amylose. So amylose is broken down by amylase. But that's the only really specific example that you must know. Moving on from there, we've got another chemical, which is bile. Now, bile is produced by the liver and it's stored in the gallbladder. And bile is a really important one in chemical digestion. It does two major jobs. The first job that it does is it neutralizes the stomach acid. There's two really important reasons for that. The first reason should be quite obvious. Okay, you need to create an optimum pH for the enzymes to work. We said in the last video on factors that affect enzymes that pH is essential for enzymes to fold into their correct shape, for their active site. If you look at the enzyme I've put way over on the far side, the active site, the mouth part, must be the correct shape to be complementary to the substrate. That doesn't happen in acids. There's one exception, which we'll talk about in a minute. But otherwise, enzymes need to be in a neutral pH, so you need bile, which is an alkaline, to neutralize the stomach acids. Okay. Of course, as well, for neutralizing the stomach acids, that protects the small intestine because the stuff comes out of the stomach with all the acid and goes into the small intestine. If it was left acidic, it could damage the small intestine. And your small intestine is where you absorb all the nutrients. So if that's damaged, you're not going to be able to absorb your nutrients. You're not going to be able to get the food to your cells. Second role that bile has is called emulsification. It emulsifies fats. Now what that means is when there's big lumps of fat, if you put bile on them, it breaks the lumps down into thousands of little droplets. Now again, that's to increase surface area relative to their volume. That exposes much more surface so that those fat droplets can then be digested by the lipase, because the fat is made of lipids. Break the fat down into droplets of lipids. The lipase enzymes, which are now at their correct pH because of the bile, can then attack the fat droplets and break the lipids down into fatty acids and glycerol. So that's a really, really important one as well, that it emulsifies fats, it breaks them down into fat droplets. That's to increase surface area to volume ratio for the lipase enzymes to work. So really important that you know all these examples of physical and chemical digestion. Really importantly, that you know all the examples of the enzymes and you know what bile does, because that comes up a lot in the exams. So let's do a quick and simple summary of this presentation. There's not huge amounts of information here. So first off, let's bring back the start diagram. Digestion, it's just polymers from the cells of other organisms being broken down by monomers. That's what digestion is. And remember that digestion is both physical and chemical. It uses both mechanisms. When we're talking about physical, we're talking about chewing and churning. When we're talking about chemical, we're talking about hydrochloric acid in the stomach, all the different enzymes, which are mostly made by the pancreas, but we'll come to that next video, and the role that bile plays. Now for enzymes, you need to know everything that's in this box. And what I mean by that, you need to know the polymer, what monomer it gets broken into, and which enzyme does that for those 4 examples. Okay. For bile, you need to be able to explain why neutralizing and emulsifying are so important to the process. Okay. Just to put it all in context, remember we had this end bit as well, that the monomers, you need them broken down so that you can absorb them, transport them, and then use them to build your own polymers in your own cells. But that last bit that I've just put up, that's the why we do digestion, to get the monomers to rebuild them as our own. But if we're just studying digestion, you really need to be more concerned with just what's inside that yellow bubble there of the how digestion occurs. Now, of course, the main thing that does digestion are the enzymes. That's the main biochemical. You need to know next where those take place and how the whole system is adapted to get the most out of the food that you eat. So that's where I want you to go next. So head straight away to digestion 2, which is on the digestive system. That's really, really important and completes the digestive system unit. Please do subscribe to my YouTube channel if you haven't done so. Like this video if it was useful. And remember, I've got the Facebook channel now. Have a look on there. There's loads of useful stuff. Thank you.